again, I wanted to elaborate a little bit more about specifically the process in eukaryotes of alternative splicing. So I already went over this slide, but I'm coming back to it. So this is showing transcription. So transcription is where one side of the DNA, specifically a side that is called the template strand or the antisense strand, is copied by an mRNA. This would not be the entire chromosome. This would be one gene out of thousands of genes that are on a particular chromosome, one single gene, a recipe, so to speak, for a particular protein that the cell needs. Okay? And so this DNA opens, the mRNA makes a copy, and in a prokaryote, if this was a prokaryote, that would be it. The mRNA would make a copy, and technically, that mRNA could already go to the ribosome while it's still being made, and it could already be going through that second step of translation. So the tRNAs could come in, and a protein, a polypeptide, could actually be synthesized at the same time that this mRNA is being built from the DNA. But in eukaryotes, I explained that there was a processing step after this. And this slide, I already went over it, but I just want to explain it again. So in prokaryotes, that's it. It gets to the terminator, the mRNA is complete, and technically it can already be going through a ribosome and manufacturing a protein. But in eukaryotes, it's going to get this 5' cap and a poly A tail. These are things that are going to help it exit the nucleus, help ribosomes to recognize it, keep it from getting broken down by enzymes, because that mRNA eventually is going to get broken down by enzymes. And then it's going to get cut, and introns are going to be cut out, and exons are going to get spliced together by these special spliceosomes. But one of the things I briefly mentioned in the previous lecture was that alternative splicing could happen to make different proteins. So I just wanted to clarify, because there was a particular AP classroom question about this, and two of the choices sounded very similar, so I want to make sure that it's clear. So this diagram is showing the introns and exons. This is the DNA. The introns are always, 100% of the time, going to get removed. Introns do not turn into exons. That's not what alternative splicing is. So introns are going to get cut out of this primary mRNA. This is a DNA, but it would have made all of this into RNA. The introns will get removed, and then the exons will get spliced together. This picture shows it also. It's showing how the introns getting removed here, and the exons are spliced together. This is the poly A tail, and this is the 5' cap. Okay, so let's talk about alternative splicing. So in this picture, these green pieces are the introns on the gene. So the gene gets copied to mRNA, including the introns. And now, on this mRNA, when it goes through the processing step, introns get cut out, no matter what. They are introns, they are nonsense pieces, they are not going to be used. But these are the exons, and they're numbered 1 through 4. So what we're saying is, in alternative splicing, if you need, say, protein A, you will selectively splice together, your cell will, 1, 2, and 3. And it'll ignore 4. 4 will get recycled along with the introns. But if you need protein B, then you're going to take 1, 2, and 4, and 3 will not get used. So that's what alternative splicing is. You can have one original gene, and then depending on which exons are selected, you can make multiple proteins, similar proteins. They may all be enzymes that have very similar structures, for example. But again, introns do not get turned into exons. I just wanted to make that super clear, that the introns get cut out either way, and then selectively the cell may choose only certain exons to be part of the protein.